Good morning, everyone. Thank you for joining us on this snowy Tuesday morning. I hope everyone is keeping warm. So today is our next in the series of the Viruses and Vaccines seminar. My name is Helen Chu. I know most of you. I'm an infectious disease physician at University of Washington. And I'm really delighted today to welcome Jesse Bloom here to speak to us. So many of you know Jesse, both locally and nationally and internationally for his work on viral antigenic evolution. So Jesse did his undergraduate degree at University of Chicago and his PhD and his postdoc at Caltech. He is originally from Montana, but is now here in Seattle with us. And his lab researches viral evolution of SARS-CoV-2 and influenza, and has really done a lot of major work in the last 2 to 3 years to help us understand escape from viral mutations through antigenic evolution. So thank you, Jesse, for joining us today. So today is Jesse's talk, and then our next talk is going to be from Keith Klugman. Let me see if I can advance the slide. And that will be, I believe, on January 17th, which is the next, on January 17th from 9 a.m. to 10 a.m. Keith is the director of the pneumonia program of the Bill and Melinda Gates Foundation. Will be speaking to us about vaccines for pneumonia and global rollout. So I'm going to stop here and turn it over to Jesse to give us talk. So thank you, Jesse, for joining us.
Thank you for the invitation. Okay, I'm hopefully sharing my slides and everyone can see them. And in one sec, I'll try to put the link. I'm going to just put the link to my slides in the chat in case anybody wants that. So if you don't want to pay attention to the whole thing, you can just go look at the link to the slides. Okay, so I'm going to be talking about interpreting the evolution of SARS-CoV-2. So let me get going here. Okay, so just my disclosures. I'm on the scientific advisory boards of A Priori Bio, Arium, Invivid, the vaccine company, and Oncorus, and I am an inventor on Fred Hutch patents related to deep mutational scanning of viral proteins. So I'm going to break my talk today into 3 parts. First, I want to talk about some conceptual principles of how viruses evolve to escape from antibodies. Next, I want to talk specifically about this important part of SARS coronavirus 2 called the receptor binding domain, or RBD. And finally, I want to understand how we can sort of interpret and understand the ongoing evolution of this protein domain. Okay, so first, some general background on viral antigenic evolution. So this is a treatise written a long time ago now by Peter Ludwig Panem, who was a Danish physician, and it's entitled Observations Made During the Epidemic of Measles on the Faroe Islands in the year 1846. And so if you read this treatise, the first dozen or so pages sort of just describe the Faroe Islands, which are sort of these beautiful, harsh, windswept islands in the North Atlantic off the coast of mainland Europe. In particular, in 1846, there was relatively limited travel from mainland Europe to the Faroe Islands. And so in the treatise, Peter Panem describes why he'd gone to the Faroe Islands, which is because measles had not prevailed on the Faroes since 1781, and then it broke out early in April 1846. And so what Peter Panem did is he went around the Faroe Islands and observed what happened during this outbreak. And he noted that of the 7,782 inhabitants of the island, about 6,000 of them came down with measles during this outbreak. But what he also noted, which made this really a classic study, which is of the many aged people still living on the Faroes who had measles in 1781, so 65 years previously, not one was attacked the second time. And so what Peter Panem is really describing here is immune memory, which is this process which provides us with lifelong protection from measles. However, as I think many of you are aware, we don't have lifelong immunity to all viruses. So for instance, the typical person is infected by H3N2 influenza probably about every 5 years. So why? And broadly speaking, you can sort of imagine two classes of hypotheses. One is that the immune response to something like influenza or SARS-CoV-2 is just not as good as the immune response to measles. So it's sort of fundamentally a deficiency in your immune response. The second one is that maybe the immunity generated to these viruses is just fine, but the viruses are evolving in a way that escapes from that immunity. And obviously, these two hypotheses are not mutually exclusive. So to try to understand, the relative role of these factors, we can sort of look at this natural experiment that happened in the history of influenza that's sort of like Peter Panham's study of measles. So if you look at the history of human influenza, the first outbreak for which we have molecular details was this flu outbreak that happened in 1918. And that caused this really devastating pandemic. It was actually even worse than the COVID-19 pandemic. Then after 1918, that H1N1 flu virus did not go away. It kept evolving a little bit every year to year and reinfecting the typical person probably about every 5 years. And that continued up until 1957, when there were some new genes from an avian flu virus that jumped into humans and caused a new pandemic, the H2N2 pandemic. And then that virus kept circulating in humans, evolving a little bit year to year and periodically reinfecting people. Then in 1968, there was another one of these pandemics, the H3N2 pandemic, and that virus has been continuing to evolve a little bit year to year in humans up until the present. So in all of these cases, we can see that people are being reinfected, but we don't really know if it's sort of a problem with their immunity or if it's because the virus is evolving a little bit year to year. But then in 1977, there was sort of an unusual thing that happened where a virus that was genetically identical to one that had existed in about 1953 or 1954 reappeared in the human population. And it's thought that it was sort of an inadvertent release of a virus that had been frozen in a freezer. And so in this case, we now have this time that's elapsed. So 23 or 24 years of time has elapsed, but there's been no evolution of the virus. So we can now ask what happened in this 1977 influenza pandemic. So here's an article describing the 1977 influenza pandemic. It was published on March 4th, 1978 in the British Medical Journal, and it's entitled Influenza in a Boarding School. So this pandemic sort of originated in Asia, and they describe how one boy from Hong Kong came back with a fever in mid-January. Within a few days, there were several boys who had the fever, and overall, 67% of the boys in this class got sick enough that they had to spend between 3 and 7 days away from class. However, they also described that of the about 130 adults at the school who had some contact with the boys, only one of them, a house matron, developed similar symptoms. So what I think this historical example is showing is that influenza infection actually also elicits multi-decade immunity, which is similar, if not maybe quite as strong as that we get to measles. But importantly, that immunity is only going to last for multiple decades if the virus is evolutionarily frozen. And although that was the case in 1977, usually these viruses are not evolutionarily frozen. So based on this sort of information, I like to think of human RNA respiratory viruses as existing on this sort of spectrum of the rate of antigenic evolution, so the rate they evolve to escape from antibodies. At one end of the spectrum, we have viruses like measles, which never really evolved to escape from antibodies. On the other end of the spectrum, we have viruses like influenza, which evolved fairly rapidly to escape from antibodies. And obviously, why these viruses fall on different ends of the spectrum is a really profound question that's sort of beyond the scope of this talk and is not totally known. But what I do want to talk about is sort of where do coronaviruses fall on this spectrum? And in particular, those of you who remember back to 2 3/4 years ago may remember that early in the SARS-CoV-2 pandemic, many people were sort of optimistically speculating that coronaviruses would be over here on the left end of the spectrum. So for instance, here's an article from the Washington Post, March 4th, 2020, entitled, "The Coronavirus Isn't Mutating Quickly," suggesting a vaccine would offer lasting protection. And sort of the rationale that the Washington Post and the scientists quoted in the Washington Post article used to arrive at this conclusion is that although all RNA viruses like measles and influenza and coronaviruses have high mutation rates, coronaviruses are the only RNA virus that have a proofreading activity in their polymerase. So although their mutation rate is still very high, a coronavirus has a mutation rate that's not quite as high as that of measles and influenza. I can hope you can recognize, however, that this is a pretty shallow form of reasoning. Evolution involves more than just mutation. Mutation is one component of evolution, but the most important thing is what is the effect of those mutations and how does selection act on them? So we wanted to really try to study this in a more nuanced way than just looking at the mutation rates. So we initially decided to study another human coronavirus called coronavirus 229E. So coronavirus 229E was first isolated from medical students at the University of Chicago in the 1960s as a causative agent of common colds. It's been circulating in humans for a very long time. We don't really know when it entered humans, but it's one of several so-called common cold coronaviruses that are responsible for about a third of all common colds. So the first thing we did is we just went to GenBank and we downloaded the spike sequences from all coronavirus 229Es. And so recall the spike is the protein that's on the surface of these coronaviruses that's recognized by neutralizing antibodies. So here's the phylogenetic tree of the coronavirus 229E spike. Older sequences from the early 1980s are here on the left. Newer sequences from the last few years are here on the right. And one thing that I hope you can appreciate, which I'll return to a little bit later in the talk, is that this tree has sort of a ladder-like shape. So there's one major dominant trunk lineage, and then a lot of short branches that come off of that. So when a virus has a phylogenetic tree of this shape, where you are on the x-axis, so where you are in time, is a pretty good indication of where you are in the evolution of the virus. So what we just did is we picked viruses at 8-year intervals across the tree, these viruses shown here in black. We synthesized those spike genes, and we generated spike pseudotype lentiviral particles that we could study in the lab. We then did a really simple experiment. We took an old human serum sample that had been collected in 1985, and we asked how well could that human serum sample neutralize viruses with spikes from each of these years. So the y-axis here is the neutralization titer. So a higher number means better neutralization. And I hope what you can appreciate is that this serum sample from 1985 can neutralize the 1984 virus quite well. But as the virus evolves into the future from 1984, the evolution of that spike is eroding the antibody neutralization. So this is showing that coronavirus 229E undergoes antigenic evolution, similar to influenza virus and different from measles virus. And I just want to emphasize the effect that's being shown here is entirely due to viral evolution because it's the same serum sample. There's also another type of measurement that immunologists sometimes like to make called antibody durability. There you take samples of serum from the same person over time, and the sample, when the sample is collected, is changing, and you're testing it against the same virus. And sometimes you can also have decreases there, and that's an immunological phenomenon. But since this plot is just showing the same serum sample against all viruses, everything shown here is just due to the virus's evolution. One other point I want to make is we've done these experiments with lots of human serum samples now. And although you always see that the evolution of the virus is eroding the antibody neutralization, the rate at which that occurs differs from serum from different people. This person's serum neutralization is very rapidly eroded by viral evolution. Here it's sort of gradual, and here it happens, but it's taking decades. And I think this is really interesting because one thing that people are sort of interested in from an applied perspective right now is it possible to make coronavirus vaccines that are more resistant to viral evolution? And this is showing that some people naturally develop coronavirus antibodies, through some series of exposure histories that stand up quite well to viral evolution. So I really think understanding the basis of these differences could be of practical value. Okay, so what I've told you so far is that respiratory RNA viruses exist on sort of the spectrum of the rate of viral antigenic evolution. And coronaviruses, or many coronaviruses anyway, such as coronavirus 229E, which I just described, and also coronavirus OC43 and SARS-CoV-2, which I haven't yet discussed, sort of live on this rapidly evolving end of the spectrum. So I'd now just briefly like to talk about the patterns of how these viruses evolve and what that implies for a vaccine strategy. So I showed you that the coronavirus 229E evolves with a ladder-like phylogenetic tree. What that means is at any given time, there are several different variants, but all of the variants die out except for one variant which wins out and then spawns the descendants. And this process keeps going and gives rise to a phylogenetic tree that looks like this. This is also how human influenza A virus evolves. And this is typical of a virus that's changing rapidly over time, but has relatively low standing genetic diversity. And when a virus evolves in this way, it's at least theoretically possible to pick a single well-matched vaccine strain because at any given time, the diversity of the virus is not that high. Coronavirus OC43, a number of decades ago, split into two lineages, each of which now evolves in a ladder-like fashion. And as some of you may know, this is also how influenza B evolves, or at least evolved until very, very recently. And when a virus evolves in this fashion, it's theoretically possible to pick a well-matched bivalent vaccine. You need a component of the vaccine that hits sort of each of these major clades. And then finally, it's of course possible for a virus to have a non-ladder-like tree where there's lots of genetic diversity that all persists and coexists at the same time. HIV would be an example of a virus like that. And if a virus evolves in that way, you're basically going to be in trouble if you're trying to make a vaccine. So how does the evolution of SARS-CoV-2 look so far? So here's just the next strain, sort of global phylogenetic tree of SARS-CoV-2. And you can see that SARS-CoV-2 has exhibited the following patterns. There have been emergence of variants which have sort of spread and become very widespread. So sort of early on, there was this variant alpha, which became very abundant. But before alpha could spread around the whole world, there was another variant called delta, which spread, and then delta became very widespread. But before delta could fully take over, there was a variant called Omicron, first Omicron BA1, then BA2, and then descendants such as BA5 that have spread very widely. And if you look at the sort of frequency trajectory, you can see there's this competition. As the amount of alpha comes up, it's pushing other things away. Then delta comes up and delta almost pushes everything else to extinction. Then BA1 becomes dominant. Then it gets replaced by BA2. Then that gets replaced by BA5. And now there's other lineages that are coming up. So we're sort of seeing this pattern of strain replacement. But the other thing I'd like to emphasize that's interesting about this tree is that one of these lineages, which is currently dominant, Omicron, exists on this very long branch. So the first observed Omicron sequences are around here. And to find the closest known relative of those, you have to trace all the way back the tree to like over here. So basically there was all of this evolution, lots and lots of evolution that happened with nobody observing it. And that's really surprising because people have been sequencing a lot of SARS-CoV-2. So how did all this evolution happen without anybody seeing it? Basically, how did Omicron acquire so many mutations with no sampled evolutionary intermediates? So we can try to understand how Omicron evolved using a principle from evolutionary biology called the molecular clock. So the molecular clock is an idea that was first put forward by Emil Zucker-Kandel and the famous chemist Linus Pauling at Caltech in the 1960s. And it's basically the idea that if you look at a sequence, the number of mutations should be proportional to how long the sequence has been evolving. So here what I'm showing is all of the pre-Omicron SARS-CoV-2 clades. I'm plotting how many mutations they have relative to an early virus from Wuhan versus when they first emerged. So you can see that prior to Omicron, SARS-CoV-2's evolution was following this nice molecular clock. But now what about Omicron? Well, if we look at Omicron, we can see that the Omicron clades are all a little bit above this line. So Omicron has extra mutations based on what you would expect from the molecular clock. So we can now try to ask, what are the characteristics of the extra mutations in Omicron? So here I'm showing all of the mutations across the SARS-CoV-2 genome. If we look at just synonymous mutations, we can see that Omicron kind of falls about on the line for synonymous mutations. There's more spread because the numbers are smaller here, but the Omicrons are not consistently above or below the line. So this shows that Omicron did not evolve due to hypermutagenesis or being a mutator strain, because if it had a higher mutation rate, it would have more synonymous mutations. We can also look at the number of amino acid mutations everywhere except in the spike protein. So about 90% of the genome codes for things other than spike. And if you look at the non-spike proteins, you can see that they also have about the expected number of mutations. So nothing unusual there. All the unusual features of Omicron relate to the spike amino acid mutations. If we look at just spike amino acid mutations, we can see that Omicron is way above the line. It has almost twice as many mutations in the spike amino acid mutations as you'd expect for a virus that age. So that fact tells us a lot about how Omicron evolved, because it's known that excess antibody escape mutations in spike are characteristic of viruses that evolve in chronic human infections. And just to explain that, let's think about what happens during the typical SARS-CoV-2 infection. There's some individual, such as this individual here, that gets infected by the virus. The virus will start to generate mutations as it's replicating in that person. However, it takes a while for any of those mutations to spread and go to high frequency or fixation, even if the mutation is beneficial for escaping antibodies. And because the typical SARS-CoV-2 infection is relatively short, and the person's typically transmitting the virus within the first week, any of those beneficial mutations haven't yet gone to high frequency. And transmission of respiratory viruses involve these very narrow transmission bottlenecks. So typically just one or a few viruses are transmitted to other people, and it's largely random which viruses are transmitted. So this disrupts selection for antibody escape mutation and slows down the rate of fixation of amino acid mutations in spike. In contrast, occasionally people experience chronic infections with SARS-CoV-2. This often happens, for instance, in individuals who may be immunosuppressed or immunocompromised. These infections can then last for many hundreds of days. So when a beneficial antibody escape mutation appears, it can then have plenty of time to rise to high frequency and even go to fixation in that patient. And this process can happen repeatedly. And so it's been observed that patients with chronic SARS-CoV-2 infections will sometimes accumulate as many as a dozen mutations in spike, real enrichment of mutations in spike. And this is probably what contributed to the evolution of Omicron, because this is consistent with the pattern we see in Omicron. Okay, and then the final point I'd just like to make is these first 3 years of SARS-CoV-2 evolution might be somewhat non-typical. So far, a substantial amount of the evolution has been driven by increases in inherent transmissibility, as well as immune escape, although now the evolution is increasingly driven by immune escape. And we know that viruses sort of eventually plateau in their inherent transmissibility, and then they just evolve mostly to escape immunity. So some of the increased transmissibility aspects of SARS-CoV-2's evolution may be transient features of the first few years. Okay, so that sort of ends the first part of my talk, conceptual principles of viral antigenic evolution. I'll just pause for a minute. If anybody has any questions on that first part, feel free to unmute yourself and ask them. Otherwise, I'll go on to the next part. Okay, I'll go on to the next part then. So
Yeah,
now,
I have, sorry, I have two questions. There was also a question in the chat, so I thought you were going to read that out.
oh yeah, sorry, I haven't been looking at the chat well. So let's see. Go ahead and ask your question first.
Yeah, so both of them have to do with the conceptual part. That's why I'm asking them now. So the first question is a more general question. So why is it that these new mutant strains, they outcompete each other? Why is it that one individual cannot be infected with two strains simultaneously? So that's I mean very general, because you showed that in
this is a really great and really profound question. So a lot of human RNA respiratory viruses like SARS-CoV-2 or coronavirus 229E or influenza A have this dynamic where one variant is outcompeting the other, which is what gives rise to the shape of this phylogenetic tree. So the explanation is thought to be as follows. First of all, these viruses transmit very rapidly around the globe. So there's not a lot of geographic structure, right? When a new variant arises in one country, within a month or two, it's in every other country as well. But then the key thing is, the idea is that there's some cross-reactivity among these different variants. So for instance, if you get Omicron BA2, you're less likely to get Omicron BA1 because there's cross-reactive immunity between Omicron BA2 and BA1. So that creates this competitive dynamic, which every one of these viruses is able to spread faster, is able to sort of use up the susceptible hosts so that the other variants don't do as well. And so that leads to one variant winning out, and then this process just continues. The thinking is that sometimes you'll get these splits where if you get two variants, and this happened for coronavirus OC43 or flu B, that manage to get far enough apart in antigenic space so that they're not competing as much anymore because there's less cross-reactive immunity, then the strength of that competition is lower and they can both coexist. And then obviously you can also get a lot of coexistence if you have a virus like HIV, which transmits very slowly, because in that case, even if you get a beneficial variant, it will take so long to spread through the population. That's kind of the best explanation. It's like a lot of things in evolutionary biology and epidemiology. It's really hard to prove what the answer is, but that's probably what it is. You had a second question as well?
Yeah, second, that's a bit more specific. To what extent is the emergence of new strains dependent on the effective population size? Because if it's only the people who are who show chronic infections, which lead to the emergence of these new strains, then that's why I'm asking this question.
So it's certainly not only chronic infections that lead to the emergence of new strains. For instance, BQ.1, which is a subvariant of BA5, which is widespread now, clearly evolved sequentially, so during transmission in the population. Some variants like Omicron and Alpha probably emerged during chronic infection. So it's not only chronic infections which contribute. I don't think it's well understood exactly how well the effective population size of the virus affects the rate of evolution. But I would say that at this point, the beneficial mutations that have been observed in new SARS-CoV-2 variants appear to arise independently many different times, which suggests to me that the evolution is more being limited by the time it takes beneficial new variants to sort of spread than by how long it takes them to emerge. So that would suggest to me that right now it's not that limited by the effective population size, but I don't know for sure.
Okay, thank you very much.
And then if someone had questions in the chat, I'm not really able to track the chat while I'm talking. So if you
Jesse,
have
this is Wes Van Voris. You mentioned that spike evolution occurs during chronic human infection, but why formally couldn't it be as yet not observed chronic animal infection?
It could be chronic animal infection. We can't rule that out. That said, so far, and there's a nice paper by Lin-Fa Wang on this, in general, the rate of antigenic evolution, which largely composes evolution, the spike, is usually slower in other animals than in humans. And this is definitely known to be the trace for influenza virus. And the reason for that is the extent of antibody pressure on a virus basically depends on the age of the host population, right? If everyone in the host population is 1 year old, most people don't have any pre-existing immunity and the virus doesn't really need to evolve antigenically. If people in the population are living to be 80 years old, then the virus has to undergo antigenic evolution to reinfect people. And so, for instance, it appears that the evolution of SARS-related coronaviruses is actually slower in some other animal species, such as bats, than it appears to be in humans. And that would be compatible with the idea that humans are longer lived than almost every other animal species out there. That said, we can't formally rule out that it could have happened in an animal, but so far nobody's observed any dynamics similar to human chronic infections happening in animals.
Thank you.
Any other questions on the first part? Okay, in that case, I'll go on to the next part. So now I'd like to get down to the details of the virus and talk about where the evolution is happening. So first, if we look at coronavirus 229E, here I've plotted in blue the sites in the coronavirus 229E spike that have changed over the last 4 decades and the extent that they've changed. So you can see that most of the change is happening in this first part of the spike called the S1. Some of it's in the N-terminal domain, but it's especially happening in the receptor binding domain, or the RBD. And now if we look at the evolution of SARS-CoV-2, so here I'm showing the mutations in one of the newest variants, Omicron BQ.1.1, relative to Wuhan HU1, you can again see that most of the mutations are in the S1 domain, and they're especially concentrated in the receptor binding domain. So it seems like evolution is really strongly putting selection for mutations in the receptor binding domain. We can also make an experimental measurement where we can ask what part of the spike are human neutralizing antibodies targeting. And so here I'm showing data where we take human sera and we deplete just the fraction of the antibodies that bind to the receptor binding domain. And recall that the receptor binding domain is only about 20% of the spike. So most of the antibodies don't bind to the receptor binding domain. But yet, if we get rid of just those receptor binding domain antibodies, the neutralization titer goes down a lot, both for vaccine and convalescent sera. So this is showing that most of the neutralizing activity in human sera is targeting the receptor binding domain. And so I think this really emphasizes the importance of the receptor binding domain. First, we have the evolutionary observation that human coronaviruses, which are basically evolving to escape transmission blocking immunity, show the strongest selection in the RBD. So in my mind, this is sort of the virus's way of saying, hey, it's these RBD antibodies that matter the most for blocking transmission. Importantly, other antibodies and T cells still put some pressure on the virus, and in particular, they may reduce disease severity, but that's less of a selection pressure on the virus. And then concordant with these evolutionary observations, we can see that experimentally that most of the neutralizing activity is from RBD-directed antibodies, although again, antibodies to other domains can also be neutralized. And so if you look at something like Omicron, here's Omicron BA1, it has a whole bunch of mutations in the receptor binding domain. So this protein is only 200 amino acids long, and Omicron BA1 has 15 mutations. So this basically means like in one evolutionary step, this protein got, something like 7 or 8% protein divergence. And so for those of you who study proteins, you know that this is like phenomenal, right? Like the typical human and mouse protein, has about 90% identity. And Omicron got as much divergence as happens between a human and a mouse protein sort of all at once. It's pretty amazing. And so one of the things you can ask is like, how is this protein able to acquire so many mutations? Because the receptor binding domain does something really important, which is bind to receptor, and that's important for the virus to get into cells. So how does Omicron even bind ACE2 with so many mutations? So to answer this question, we've developed this deep mutational scanning approach to measure how mutations to the RBD affect ACE2 affinity. And so the way we do that is we just take yeast and we display the receptor binding domain on the surface of yeast. And then we can incubate those yeast with fluorescent ACE2. And the better the receptor binding domain binds to ACE2, the more fluorescent the cell will become. And we can do this with a big library of yeast, each of which have a different receptor binding domain on their surface. And then we can sequence these yeast to figure out which ones are binding well to ACE2. And so doing this, we can generate these very rich maps of the effects of mutations. So here I'm showing every mutation in the receptor binding domain. So like this is the mutation W353T. And we've measured how much that mutation affects ACE2 affinity. It's colored red because it's kind of bad for ACE2 affinity. In here, mutations are colored red if they're bad for ACE2 affinity, white if they don't really have any effect on ACE2 affinity, and then they're colored blue if they increase ACE2 affinity. So if you just kind of look through, you can see there's a lot of red and a lot of white, because most mutations either are bad for ACE2 affinity or don't have much of an effect, but there's some mutations that are blue. There's some mutations out there that actually increase ACE2 affinity. And this has been really useful to sort of track the evolution of this virus. So for instance, in late 2020, there was these variants that started to emerge that had this particular mutation N501Y. That mutation increases ACE2 affinity, and it's thought to have contributed to the evolutionary success of the virus. And just in general, as new mutations are arising in SARS-CoV-2, it's great to have these tables where you can just go look up what that mutation does to the ACE2 affinity. We can also look at broader trends in this, and in particular, we can look at what are the mutations that are mediating antibody escape, which we can also experimentally measure in a way that I'll describe in a minute, and the mutations that are increasing ACE2 affinity. So on this plot, I've drawn a line at 0 indicating the effects of the mutations on ACE2 affinity. So mutations above the line increase ACE2 affinity, and mutations below the line decrease ACE2 affinity. So hopefully what you can appreciate is all of the mutations that are sort of shifted to the right here, so that mediate antibody escape, are bad for ACE2 affinity. And the reason the virus has been able to get these mutations is it's also getting the mutations above the line here, which don't have much effect on antibody escape, but increase ACE2 affinity. And there's sort of this buffering going on. These mutations are helping enable these mutations. And importantly, I don't think this process is going to stop. So if you look at something like coronavirus 229E and its receptor binding domain, 25 of the 31 residues that contact the receptor have varied over the last 50 years. So like almost all of them. And we can just see experimentally, there's lots of mutations to the SARS-CoV-2 receptor binding domain that retain or even enhance ACE2 affinity. So I think this process of the virus accumulating mutations that erode the neutralization by these antibodies is just going to be what happens for the foreseeable future. Okay, so that is sort of the summary of the receptor binding domain. I'll pause again. Does anybody have any questions on that before I get to antibody escape? Okay, so the next thing I want to talk about, which I think is probably of the most interest to people, is how this virus is evolving to escape antibodies. So we can also measure that in very high throughput experimentally, just by putting this receptor binding domain on the surface of yeast, and then seeing which receptor binding domains have mutations that escape antibody binding. And so, for instance, here's what happens if we do this for one particular monoclonal antibody called LY-CoV555 or bamlanivimab. And here I'm just summarizing the data at the level of sites. So you can see that this antibody is escaped by mutations at sites like 452, 484, or 490. So the immediate implication of this plot is simply that bamlanivimab was a very unfortunate choice of an antibody for Eli Lilly to develop as a therapeutic, because the Delta variant had a mutation at site 452, and most other variants had a mutation at site 484. So this antibody was virtually useless almost as soon as it got an emergency use authorization. So obviously one thought would be, as if you're developing monoclonal antibody therapeutics in the future, it might be useful to prospectively identify these escape mutations and at least try to choose an antibody that didn't seem to be targeting sites where the virus is undergoing rapid evolution. But more generally, what I want to talk about is what antibody escape applies for the polyclonal antibodies that are elicited by infection or vaccination. Because again, it's really natural immunity or vaccine-induced immunity that's protecting most of the world out there, and not a monoclonal antibody therapeutic. And just to emphasize the difference, a monoclonal antibody targets one region of the virus. So if the virus gets a mutation, the antibody doesn't bind anymore. However, the human immune system makes a polyclonal antibody response, which basically consists of a whole bunch of monoclonal antibodies. And if those monoclonal antibodies in the polyclonal antibody response are targeting many distinct epitopes, then even if the virus escapes one of them, there's still going to be a lot of other antibodies which can neutralize it. So now I just want to kind of give a little toy example of how we use our experiments to look at this process. So here's a hypothetical polyclonal antibody response, which consists of just these three different antibodies, the pink one, the purple one, and the blue one. And for each of these antibodies, we've measured which mutations affect antibody binding. And so for the mix, I'm just averaging them together to get the black line, which sort of tells what this polyclonal mix is targeting. Now you can say, what happens if the virus gets a mutation at site 484? That's going to knock off the purple antibody. So now the black line looks different. And I want to make the point that getting a mutation at site 484 is also escaping the antibodies that target site 490. So the peak at 490 goes down as well, because it's the same antibodies that target those two sites. They're in the same epitope. So that's basically the principle. If we have these data for lots of monoclonal antibodies, we can just aggregate them together. And the reason this has been powerful is that Tyler Starr and Ali Greeny in our group sort of developed this approach and applied it to several dozen antibodies, which started to give us a good data set of what the human immune system was targeting. But then really remarkably, the group of Yunlong Cao, Suni Shi, and colleagues at Peking University has sort of industrialized this approach and applied it now to over 3,000 human antibodies. So we now have this huge set of antibodies where we know all of the escape mutations. And so that forms the basis for our antibody escape calculator, where what I've done is I've just taken these 3,000 antibodies and I've aggregated them together to see what they tell us about the sites that are being targeted by the human antibody response. So here I'm showing antibodies that neutralize Omicron BA2. And so it turns out that this sort of escape calculator gives us a lot of power to understand and to some extent even predict what happens. So we have this data for Omicron BA2. If you look, you would see, all right, in the background of Omicron BA2, the virus could escape these antibodies by getting mutations at sites like 346, 444, 452, 486, et cetera, those big peaks. So no, what happened after the BA2 wave? There was then a new virus that emerged called BA5, and BA5 had a mutation here and a mutation here. So it's sort of taken two of these peaks, which knocks off a substantial fraction of these antibodies. Then there was this BA5 wave. Then what happened after that? There was the emergence of another variant called BQ.1, which had now picked up a mutation at site 444, knocks off more of those antibodies. Now BQ.1 has spawned descendants, the most successful of which is one called BQ.1.1, which has now also picked up a mutation at the highest remaining peak, 346, and et cetera. And now there's viruses out there that have mutations at 490 and more mutations at 484. And it's kind of uncanny. It's almost like the virus is just looking at these experimental data of which mutations cause the biggest neutralizing antibody escape and getting mutations at those sites, which I think really shows how strongly it's escape from neutralizing antibodies that is currently driving the emergence of these variants. So that is sort of the antibody escape calculator. I have some slides that just show what I just went through interactively. The last thing I would like to talk about is what about going forward? So one of the things that we would really like to be able to do is keep tracking and understanding viral evolution. And I described how we've been doing this using this yeast display system. And that has some advantages. It's safe, it's high throughput, et cetera, but it also has some limitations. It only examines mutations in the receptor binding domain, which is just part of spike. It only measures antibody binding and not neutralization. And this is important because it seems to be pressure to escape neutralization that's really driving viral evolution. And it only works for viral entry proteins with domains amenable to yeast display. So to overcome this, we've been developing an alternative deep mutational scanning system that's based on lentiviral pseudotyping. And the advantage of lentiviral pseudotyping is that many viruses have entry proteins amenable to pseudotyping, and it provides a safe way to study entry proteins from viruses spanning coronaviruses to influenza viruses to Hanipa viruses and all these different things. However, unfortunately, traditional pseudotyping does not create a genotype-phenotype link. So it's not immediately translatable to deep mutational scanning. So, and I realize this plot may only appeal to the aficionados, but we've developed a way to get around that by developing a lentiviral system where we can link the genotype of the lentivirus to the spike protein on the surface. And to do that, we encode the spike in the lentivirus, and then we use a two-step process to make cells that are expressing just one of these lentiviral genomes, and then use that to generate a viral library, in this case, pseudotype with spike. The other key innovation in this system is that sequencing, which is what you can do for these libraries, only measures relative amounts, but neutralization is an absolute measurement. So to get around that, we include in our library a non-neutralized standard, such as VSVG, which is not neutralized by human sera. We can then sequence all of this and convert our sequencing counts to absolute neutralization using that standard. And so using this approach, we can now take any human monoclonal antibody to SARS-CoV-2 that binds anywhere in the spike and figure out what are the escape mutations. So for instance, here's the antibody LYCO1404, which was one of the only antibodies that, at least until recently, neutralized SARS-CoV-2. So we can very quickly map out where its epitope is. It's in the receptor binding domain, and we can zoom in on the key sites that escape binding by this antibody and understand how it's going to be affected by future mutations. And because this high throughput assay is measuring directly neutralization, the estimates we get of how mutations affect neutralization correspond very well to what we measure in conventional neutralization assays. So we get this great correlation. Importantly, we can also apply this method, unlike the yeast display, to antibodies targeting other regions. So for instance, some people are currently interested in coronavirus antibodies that target the S2 domain, because some of those antibodies can cross-neutralize not only SARS-related coronaviruses, but even MERS-related coronaviruses. So for instance, here's one of those antibodies, and again, we can map out, its epitope. We can see it's in the S2 domain, and we can even zoom in and really figure out exactly what mutations affect neutralization by that antibody. So I think this is potentially a really powerful approach to generalize our ability to study viral antigenic evolution. So just to sort of wrap up, a subset of human respiratory viruses evolved to escape immunity. SARS-CoV-2 is unfortunately in this subset. The most important antigenic evolution occurs in the receptor binding domain. We've been able to use deep mutational scanning to identify the antibody escape mutations in the receptor binding domain prospectively, which is useful for various purposes. And we're now developing approaches that it can extend this both to the full spike and the entry proteins of other viruses. And so we think, again, this could provide, because we know for both coronaviruses and other viruses, it's really the entry proteins that are the important immunogens. We think the ability to really comprehensively understand how mutations affect those in this sort of safe non-viral context or non-replicative viral context could be really valuable for antibody and vaccine development. And finally, I'd like to end with some thanks. I'd like to thank Tyler, who's recently started his own lab at Utah, and Ali, who did most of the deep mutational scanning I described. Rachel did the work on coronavirus 229E, and then Bernadetta, Kate, and Kaylin have developed this new lentiviral deep mutational scanning system that I was describing. And I'd like to thank a lot of great collaborators, both here at University of Washington and the Hutch and elsewhere. And I'd be happy to take any questions. If anybody's there, if you have a question, just go for it.
Hi, Jesse.
Hi.
Great talk. I'm curious about, so these are single mutations one at a time, but of course you showed with Omicron these 15 mutations in the RBD. And is there any add-on effects when you're when you're changing the RBD that dramatically in terms of ACE2 binding or antibody escape?
Yes, that's a great question. So there's sort of so within the receptor binding domain, we have done some studies that have tried to look at sort of epistasis or the effects of multiple mutations on ACE2 affinity. And we find that there is a little bit of epistasis. So actually, it turns out most of the mutations have the same effect in all the different variants. But there's one specific pair of mutations, N501Y and Q498R, which together have a very synergistic effect on ACE2 affinity. And that's played a very important role in sort of the emergence of Omicron. Another form of epistasis, which we are working on studying in this lentiviral system, I didn't really have time to elaborate it here, but our papers sort of describe it, is looking at epistasis in antibody escape. And there, the epistasis can simply arise from the fact that there are multiple epitopes. So let's say the biggest portion of your neutralizing antibody response targets epitope 1, but there's also some antibodies that target epitope 2. If you mutate epitope 2 alone, you don't see much of an antigenic effect because most of the antibodies are targeting epitope 1. But if you mutate epitope 1, then your neutralization goes down because then you only have the antibodies to epitope 2 left. And then if you mutate epitope 2 and epitope 1, you get an even bigger drop in neutralization. So that's another form of what I would call antigenic epistasis, and we are trying to work on ways to study that. So it's a great question, and epistasis is a really important thing to look at here. And we're limited by the fact that we can enumerate the single mutants, but we sort of have to be clever to study the multiple mutants because there's so many of them.
That's great, thanks.
I got the question from Jody about the Lin-Fa Wang paper. I will try to share it in the chat, but I would also say just send me an email if you get disconnected from this before I do it, because I can find the paper, but I can't find it off the top of my head.
Great talk, Jesse. Could you say what lentiviral pseudotyping is, please?
Sorry, what about viral pseudotyping?
What is the definition of lentiviral?
Oh, yeah, so a lentiviral pseudotype. So basically, a lentivirus is HIV, and what people have done is they've taken HIV and stripped it down, so they've gotten rid of the proteins that actually needs to be infectious. But you can put a reporter in there, like you can put GFP. So then a lentiviral vector is sort of a stripped down version of an HIV, which can only infect a cell once, and so it's relatively safe, at least for research purposes, that will make a cell sort of indicate that it's been infected. So what pseudotyping now means is you can take that stripped down lentiviral particle and you can put the entry protein of many different viruses on there. So for instance, a lentiviral particle can be pseudotyped by the spike of a coronavirus, SARS coronavirus, a MERS coronavirus. It can be pseudotyped by influenza hemagglutinin. It can be pseudotyped by the receptor binding protein and fusion protein of a Nipah virus, or the glycoprotein of Lassa virus, all of these different things. And this is a really powerful way to study these viral entry proteins, because you can then study them in the pseudotype format where you're not working with the human pathogen, but you're still measuring the role of this protein in terms of infecting cells. So actually the most common way, for instance, that people measure antibody titers against SARS-CoV-2 is not actually working with SARS-CoV-2, which is more difficult to work with because it's a human pathogen, but often using these pseudotype assays, which are sort of both easier and safer. And then the pseudotyping becomes, especially important if you're working on something that might even be more high risk than SARS-CoV-2, like a Nipah virus, let's say.
Hey, Jesse, great talk. I had a question about this new lentivirus system as well. I'm wondering, could you use this same system to study regions of spike that are not very plastic or not able to, mutations?
Yeah. So that's a great question. So I didn't really have time to go into that, but you can definitely see when you look at these deep mutational scanning data, there are some regions of spike that are less mutationally tolerant than others. In particular, like some of these stem helix regions in the S2 domain are not very tolerant of mutation because they do really important things for fusion. So we actually can use these methods to identify regions of these proteins that are less tolerant of mutations and are less likely to undergo change. So I think that's really valuable. That said, the trick is that the challenge is that so far it's been much easier to identify really potent antibodies against the receptor binding domain than against something like S2. So we do know, I mean, we being the field collectively, do know of some antibodies to, let's say, S2 that target regions that are pretty mutationally constrained or functionally constrained. Whether those antibodies will ever have sufficient potency to be useful as therapeutics, or whether they can be elicited to high potency by vaccines, I think remains an open question.
Okay, so with that in mind, I mean, do you have any opinions or thoughts about the current approaches to universal COVID-2 vaccines? You know, there's those that are targeting that same stem region versus those that are using an RBD approach. Do you think there's any favor in either of those approaches?
Yeah, I mean, so as you obviously know yourself, there's a whole bunch of approaches out there for coronavirus vaccines that ideally would be better. I don't have the answer to this. I mean, I certainly think that the antibodies targeting regions like S2 are less likely to be escaped. However, whether those antibodies can ever be made potent enough to provide really strong, protection against infection is unclear, because those epitopes just don't appear to be as neutralizing. So I think there's a variety of interesting approaches to try to develop broader vaccines. I also feel like an important component of vaccines, I know there's been a lot of emphasis over the last decades on sort of broad vaccines to many viruses. I also think with mRNA vaccine technologies, we should also think about fast vaccines, equally as important as broad vaccines. There's sort of two approaches to deal with both a new emerging variant of a human virus or a new pandemic virus. One is to sort of have some broad vaccine you already have, saved up. But the other one is with mRNA vaccine technology, if you can just make a new, maybe fairly strain-specific vaccine to that new variant very quickly, that could end up being also an effective approach that could turn out to work better. So I don't know, there's a lot of approaches out there. I think many of them are worth exploring. Which ones, if any, will work is something that I don't know the answer to, but that's why I think they're kind of all worth exploring.
Thank you.
Any other questions?
Jesse, I'm monitoring the chat. There are a lot of thanks for your talk.
I think I've answered some of
Yeah,
them.
but no more questions, I believe. So, and it looks like we are at time. So
Okay.
thank you, Jesse, for, hold on. Just more thank yous.
Okay, and Jody, who put the question about or whoever put the question about the Lin-Fa Wang paper in there, I'm not going to be able to find that in like 2 seconds. So just send me an email and I'll reply with it in the email.
Yeah. Great. Thanks
All right.
everyone for
Thank you,
joining.
Dawn.
Thank you so much, Jesse. Happy holidays.